Hello, my name is Chris Pinto, and I will be starting off this presentation for Group 4. Today we will be reviewing the paper, Tumor Morphology and Phenotypic Evolution Driven by Pressure from the Microenvironment, by Alexander Anderson et al. In order to fully understand the grounds of this paper, it's important to recognize what we already know about tumor morphology. Cancer is simply the uncontrolled division of cells, which is typically triggered by a mutation in oncogenes or tumor suppressor genes. Some cells can get around these mutations by performing apoptosis, while others don't and overreplicate via selective cloning. After some period of time, cancer tissue or tumors will begin to form, become increasingly aggressive, locally invasive, and ultimately metastatic. What is poorly understood is this next step in tumor evolution, which is metastasis, where the cancer starts to spread to different parts of the body, usually via the bloodstream or lymphatic system. While tumor formation is clearly regulated by genes, the selective pressure for metastasis is less obvious, since there are several different factors that are involved. There are also several discrepancies and paradoxes too that lie within delineating the conditions for metastasis. For example, lung carcinoma metastasize early, while still small, whereas other tumors such as breast adenocarcinoma metastasize as a function of size. In all, the mechanism of selection for invasive cancer cells remains an open fundamental question in cancer biology. Thus, these researchers created a multi-scale model of tumor morphology to study what triggers metastasis. Using a host of mathematical description and simulation techniques, the researchers were able to simulate a wide range of microenvironment conditions and tease out their effects on tumors. All this will be in more detail to follow. Now moving to the significance of this paper. Recognizing what environmental conditions affect tumor evolution opens up the gateway for customizable medicine. The hopeful application of this is to be able to take small samples of the tumor and its own environment, recording their respective properties, and running an accurate simulation that will tell you the future development of that tumor. With this information, you can devise a better treatment protocol, administer specialized drugs, have a custom therapy plan, and the list goes on. Such an advancement in computational biology could truly revolutionize cancer treatment and better the lives of many.
So I'm Quinn, and I'll be talking about the approaches to the study. The overall model used for the study was a hybrid discrete continuum model. Such models allowed for the input of both discrete and continuous variables, as shown on the slide. Some of the continuous variables used in this model represented the interactions between cells as well as the extracellular matrix. Some of the discrete variables represented how individual cells grew, divided, moved, and mutated themselves. All variables can be linked to various measurements established by cancer biologists. For the movement probabilities, which represent the discrete system, the cells are modeled on a 2D lattice. From there, probabilities can be calculated for the changes to various states. These probabilities depend on the intrinsic unbiased motility, which is the dn term, as well as the extracellular matrix, which is modeled by the chi and f terms. The other cellular parameters are the continuum system, which contributes to the life cycle flowchart that the HDC model depends on. These parameters involve the extracellular matrix and oxygen changes over time. The continuum system takes into account both cell phenotype and microenvironmental influences, which include basic metabolic and motility-related processes. These are the two life cycle flowcharts used in this study, which provide a natural link between the mathematics and biologic experimentation. The linear flowchart was initially used to model cancer invasion. However, in order to eliminate the phenotypic selection bias present, the random mutation scheme was composed. Both structures were proved to provide a realistic outcome for modeling.
My name is Seppo, and I will be talking about the strengths and weaknesses of this model. The big advantage with this HDC approach was that it was a hybrid model that treated some variables as continuous and some as discrete. The advantage of continuous variables is that they can be solved using partial differential equations. However, this also means that some variables such as cells are treated as an average, which would be inaccurate. Processes such as cell-cell adhesion cannot be fully characterized by a continuous model, which makes the hybrid HDC model more precise. Depending on their relative importance, individual cell traits can also be included or excluded from the model. The individual significance of cell traits' contribution to the invasive process can be determined experimentally. Other important occurrences such as cell death are also included in the model, as they can be a result of oxygen deprivation. This was crucial to consider as it affects the rate of growth of the tumor. As we can see, this model is quite robust. However robust this model may be, it however still has some weaknesses. One major weakness is that it assumes that cells have a limited number of phenotypes. Although researchers were able to define an array of random phenotypes, it is important to remember that minor mutations in the cell's genome can lead to completely different phenotypes. Depending on the characteristics of the cell phenotype, it may become more or less invasive. The effect of changing a single variable in this model is still difficult to predict and is still an ongoing investigation. Current experiments are the only way to predict the outcomes.
Now I am going to discuss some of the results of this study. Here we're looking at the effect of different ECMs on tumor morphology. Each column represents a different density distribution of the matrix, while the bottom two rows show the results of the simulation using either a random or linear mutation scheme. We can see that the tumors metastasized, or fingered outwards, in the heterogeneous matrices regardless of their mutation scheme. This indicated that the mutation schemes do not drive metastasis, but the environment itself does. Shown here are the phenotype distributions from the last slide. The primary takeaway is that fewer phenotypes survive in the heterogeneous conditions. More importantly, of the phenotypes chosen, they tend to be more aggressive. This includes lower cell-to-cell adhesion and lower oxygen consumption. These findings further support that tumors develop to be more invasive under heterogeneous conditions. Next, researchers explored what other environments may induce fingering behavior, such as one with low oxygen. Here we can see that when the oxygen concentration in the ECM was dropped, tumor fingers began to develop again. By now, it is clear that harsh environmental conditions support invasive tumor development. Similar to the ECM studies, the researchers provided the phenotypic variation in the oxygen tests over time of the simulation. Looking at the y-axis, times between 0 and 50 and 250 on were oxygen-rich, while times between 50 and 250 were not. Each flare-up depicts the amount of cells expressing one of the 100 phenotypes listed on the x-axis. We can see that for mild conditions, there's a lot more diversity in phenotypes and lots of clones coexist, while in harsh conditions, only a few dominant and aggressive phenotypes emerge, and thus form tumors that eventually metastasize. Lastly, the researchers repeated the last assay, but now added a new variable, adhesion types. By going through high and low levels of oxygen depicted in each square, you can see fingers develop, expressing different adhesion types as seen in the green and blue coloration in row B. What is remarkable at this experiment was that the HDC model developed was able to capture this complex biological behavior. Also, this showed that it can handle data on a multi-scale level, show effects at the tissue scale, and can be manipulated to introduce new variables and further our understanding of tumor morphology.
Hi, I'm Caitlin, and I'm going to be concluding with the impacts of this research. Researchers found that the type of microenvironment, whether it be harsh or mild, had a huge impact on tumor behavior. As stated previously, harsher environments led to more invasive and aggressive tumors dominated by only a few phenotypes. This could help to explain experimental and clinical findings showing that hypoxia, or nutrient deprivation, leads to aggressive tumor behavior. Also, if these invasive tumor properties are reversible under appropriate microenvironment conditions, perhaps differentiating therapy aimed at cancer microenvironment interactions may be more useful than simply making the microenvironment harsher, like by chemotherapy or anti-angiogenic therapy. The HDC model directly supports the clonal dominance theory of metastasis, suggesting that invasive cells are clones where their properties are independent from growth factors. Metastasis is then the consequence of such clones taking over and dominating the tumor. However, this is in contrast to the prevailing dogma. While the mathematical model reliably predicts the clonal theory, the debate among scientists remains unresolved. It is hard to reach clear-cut conclusions from experimental work, but mathematical simulations like this one can provide a platform to consider many variables simultaneously and help scientists dig deeper. There are still improvements that need to be made in HDC modeling, such as better representations for molecular networks or predictions of singular molecular changes, as well as the prevalent computational limitations. However, eventually these models could be used to predict invasive behavior of individual tumors based on actual measurements of cellular and microenvironmental parameters. This could be useful in drug delivery or in the management of cancer patients. For example, high-resolution imaging techniques measuring properties of cancer cells and the microenvironment, in combination with modeling, could be used to customize therapy in individual patients. This could pave the way for better and better treatments in the future.
Here is a slide with 3 questions for further discussion and also a thank you to conclude our presentation.